Hi everyone, my name is Kyle Hoban. I'm a postdoctoral research scientist and postdoctoral instructor at Johns Hopkins University, and I work in Dr. Beverly Wendland's lab in the Department of Biology. First off, I just want to say thank you to the organizers for giving me this opportunity to share some of our recent work with you. I think it's great that even in the midst of this pandemic, we're still able to gather and communicate science, even if it's online. Our lab is a cell and molecular biology as well as genetics group, and we ask some really fundamental questions about a biological process that infectious microbes like this novel coronavirus use to infect cells. And so though I'm not a virologist or epidemiologist, I'm planning to dedicate the first part of my talk to discussing what we're learning about SARS-CoV-2, the virus that causes COVID-19, and how it infects cells and replicates at the cell and molecular biology level. And then after that, I'm going to tell you two quick stories, the second of which I think will be most interesting to this audience as it has some implications for diseases like COVID-19. But the first one will be important for setting that up nicely. And I think it's pretty interesting as well. I hope you'll agree. So let me start sharing my slides with you. Both of the stories revolve around this protein VPS4 and the effect that the loss of this protein's function has on cells, including clathrin-mediated endocytosis mutants, and I'll explain what this is later. And as we get started, I'm going to stop my camera so that I'm not distracting us or covering up the slides. So here's an image you've probably seen versions of many times a day over the past couple of months. This is a diagram of SARS coronavirus 2, SARS for sudden acute respiratory syndrome. The original SARS coronavirus first appeared in China in 2002, and whereas that virus infected about 8,000 and killed around 800 people worldwide, the current global estimates from the Johns Hopkins tracker, at least as of this recording on April 30th, is over 3.2 million people infected and over 230,000 having died of COVID-19 or coronavirus disease 2019. So this is on a scale that the world hasn't seen since the Spanish flu pandemic almost exactly a century ago. And the New York Times has done a nice job of graphing the number of new cases and casualties over time. And these numbers have remained high and fairly static over the past few weeks. And what we're hearing from epidemiologists is that even if we're able to control the spread of the virus in this wave, we may see an even greater number of cases in the fall and winter. So this is an issue that we're not only dealing with right now, but we'll be grappling with for months or potentially years to come. In addition, we've learned that there's quite a range in the severity of COVID-19 cases. Even in the early days of the crisis, it appeared that less than a third of cases reported internationally required hospitalization. And a new study on the extent of the virus's spread in Iceland, which has been a leader in testing rates, particularly when compared to the size of its population, found that about 43% of those tested reported having no symptoms at the time that samples were taken. So though many of those people most likely develop symptoms later on, it does speak to the long incubation period, the period of asymptomatic contagiousness, and therefore the high potential to unknowingly transmit this virus to others. So what are some of the risk factors for having one of these more severe or fatal forms of COVID-19? One is certainly age. You can see from this graph that with each decade of life, the risk of dying from COVID-19 increases exponentially. Another is pre-existing health conditions, particularly those with heart and respiratory disease, diabetes, and now obesity is understood to be part of this group as well. And finally, SARS coronavirus 2 appears to be slightly more fatal for male patients. So why is it that COVID-19 is more likely to claim the lives of people in these groups? Well, the short and unsatisfying answer is that we don't really know at the moment. However, we're learning more at a rapid pace, and there are some current theories as to why this is. So there are several things that can impact the way our bodies respond to infections. Some of those variables are shown here and will be important for attempting to explain at least the differences in fatality rates between men and women, and these include a different set of sex chromosomes. Many of the genes important for immune responses are located on the X chromosome, and so men have lower expression levels of these genes on average, compared to women with only one X chromosome, which could help to explain why they're more likely to die from COVID-19. And it's also understood to be a reason why women are much more likely to suffer from autoimmune disorders like rheumatoid arthritis and lupus. And sex plays a large role in determining which hormones our body makes, which in turn impact biological responses to infection. And I read recently that new studies are looking at estrogen as a potential treatment for this disease. As for older populations being more susceptible to the severe forms of COVID-19, the number and diversity of a type of immune cell called T cells decreases with age. And also, antibody production is slower and less effective in the elderly. So together, this puts them at a greater risk of dying from this disease. However, we know currently that the very young seem to be spared from these severe infections for the most part, even though their immune systems are still developing and are less efficient than those of most adults. So clearly there's more going on here, and I think some of these additional ideas would be better absorbed after some more background. So I'll get back to more of these theories as to why risk factors exist and how our lab's work may help to better understand this in a little bit. But for now, let's talk more about the virus itself and its components. This virus is a lineage B beta coronavirus, and the first thing you're likely to notice about it in all of these diagrams is this outermost layer of proteins that surround the spherical viral particle. They're lighter in this transmission electron microscopy image on the left and are shown in red in this diagram on the right. Each of these is a trimer of the aptly named spike glycoprotein, or S protein, and these spikes are critically important for the virus's attachment and subsequent entry into our cells. And these spike proteins are also the targets of vaccine-induced antibodies. So the S protein is one of the most important components of the virus for medicine and for this talk. Now, the C termini of the spike proteins are embedded in the viral membrane, along with other membrane proteins shown here. And within that spherical membrane is the SARS coronavirus 2 genome, which is a single helical RNA molecule that's coded in nucleocapsid proteins, or N proteins. Now, the virus needs to get its genome into a cell in order to replicate. So let's take a look at a mammalian cell and some of the features that are important for this viral replication process. So here you're looking at a diagram of a mammalian fibroblast, which is a cell that produces collagen and other components in the extracellular matrix. And many of the most important structures within cells are labeled here. One that will be critical for this talk is the plasma membrane, or cell membrane as it's sometimes called. And the most important function of this structure is to separate the cytoplasm within the cell from the surrounding environment, defining what is inside and what is outside of the cell. Some other structures that will be important for talking about viral replication are ribosomes, which translate RNA into protein, the endoplasmic reticulum, or ER, which is a folded structure that surrounds the nucleus and where ribosomes sit and translate and incorporate membrane proteins. And then finally, once those proteins are translated, they get delivered to the Golgi apparatus. And the function of the Golgi is to deliver cellular components to their proper destination. And so having mentioned those, let's now talk about what actually happens when SARS coronavirus 2 enters a human cell. So this is a diagram of the replication cycle of coronaviruses in general, not necessarily SARS coronavirus 2. And it has some aspects that I like for the purposes of this talk. So for viruses to gain entry into the cell and replicate, its RNA genome has to escape its own membrane and get into the cell's cytoplasm. And one of the reasons I like this figure is that it doesn't go into too much detail about that first step of viral entry, because we're going to talk about that a lot more after this slide. And there are actually multiple ways by which coronaviruses can enter cells. So for now, let's jump ahead to after the viral genome has already made its way into the cytoplasm. Coronaviruses have a positive sense RNA genome, which means that immediately upon entering the cytoplasm, ribosomes within the host cell can start translating that genome into the viral polymerase enzyme. And that viral polymerase can then turn that positive strand RNA into the negative strand. And then that negative strand can in turn be used to make new full-length copies of the viral genome, as well as subgenomic RNA fragments for translating viral proteins like the nucleocapsid proteins and membrane proteins within the ER of the host cell. Now, this process probably does not occur like it's shown here. There's probably no new viral particles fully formed within the ER. And so for what happens from this point, I like this diagram more, where they're showing that viral membrane proteins are transported from the ER to the Golgi and then into Golgi-derived secretory vesicles. And then it's at this point that the coded RNA genome associates with those viral membrane proteins and begins this process of curving that vesicle inward. And eventually the two ends of that curved vesicle fuse together, forming two new complete membranes, one being the viral membrane itself within the vesicle, and then the outer membrane of the secretory vesicle itself. And so in this way, when that vesicle fuses with the plasma membrane, it allows for the release of the virus into the surrounding environment. So having said that, let's get back to the first step of viral entry. How does the virus go about getting its genome into the cell, out of its own membrane and into the cytoplasm? Coronaviruses, as I mentioned, can do that in two ways. Both methods rely on the virus attaching to the host cell through the binding of its spike proteins to a receptor on the host. And ACE2 has been shown to be that receptor in both cases. And this is angiotensin converting enzyme 2. But this is where the two mechanisms diverge. And the one on the left, after attaching to ACE2, the virus tricks the cell into internalizing it through a membrane trafficking pathway called endocytosis, which curves the plasma membrane inward, generating a vesicle within the cytoplasm that contains a piece of the plasma membrane and extracellular material, which in this case includes the virus. Now, eventually that vesicle and its contents are delivered to compartments within the cell called endosomes, and it's here that the virus can escape through the fusion of its membrane with the endosomal membrane. And in the other coronavirus entry method, this fusion of the viral membrane actually occurs with the plasma membrane itself, allowing for direct delivery of the RNA genome into the cytoplasm. And this is aided by the presence of serine protease TMPRSS2, which activates the spike protein and allows for that fusion event. So this brings us to another idea as to why COVID-19 severity ranges so widely. It's possible that the concentration of proteins important for the virus's entry, like ACE2 and TMPRSS2, may vary between these groups of people and contribute to the differences in the outcomes observed. And the work I'll show later has some implications for this. And before I move on from this slide, I just want to mention that a recent paper identified that the novel coronavirus needs to undergo endocytosis to gain entry into the cells, at least in their tests. And our lab studies endocytosis, so let's talk more about this process and how it works. And these endosomes are part of the endocytic pathway that leads from the plasma membrane into the cell into organelles called lysosomes. And to show the full endocytic pathway, I'm going to pull up this figure here, which demonstrates that once endocytic vesicles are released from the plasma membrane, they're first delivered to organelles called early endosomes. Now from here, material can either be recycled back to the plasma membrane, or if it's meant to be degraded, delivered to structures called multivesicular bodies, also known as late endosomes. And these are characterized by these intraluminal vesicles that they contain. And then finally, material to be degraded is delivered to lysosomes. Now, the Wendland lab uses the model organism budding yeast, or baker's or brewer's yeast, to study endocytosis. And that's because budding yeast also performs endocytosis. They have a lot of the same structures. For example, they also have a plasma membrane underneath their cell wall, and they have a structure similar to lysosomes called vacuoles that perform a lot of those same functions. And budding yeast is a really powerful organism for doing genetics. And each one of these cells allows you to look at phenotypes in the context of an entire organism. So now that I've promoted yeast a little bit, I also want to mention a few other reasons why you should care about endocytosis in addition to COVID-19. Endocytosis is important for cells to be able to function properly. For example, it's important for the uptake of certain nutrients like cholesterol and iron. And it's also responsible for regulating things like signaling events and the concentration of transporters and ion channels within the plasma membrane. It can also be used for things like drug delivery and gene therapies. And whenever cellular traffic is disrupted, it can lead to significant consequences for the cell. And endocytosis has been implicated in a number of diseases like cancers and neurodegenerative disorders. So it's important even outside of the context of SARS coronavirus 2. The Wendland lab has historically focused on clathrin-mediated endocytosis, or CME for short, and it involves this protein called clathrin that forms these trimers, also known as triskelions. And those triskelions can further oligomerize to form either beehive-looking flat structures on the inside surface of cells, or these soccer ball-looking cages called clathrin coats around forming vesicles. Now, there are also other endocytic pathways called clathrin-independent endocytosis pathways that don't require clathrin, and instead they rely on the functioning of these small GTPases. But for today, we're going to focus exclusively on clathrin-mediated endocytosis, or CME. Now, this diagram kind of outlines the major steps in the clathrin-mediated endocytic pathway. And to just briefly go through this, it involves, initially the recruitment of these early arriving endocytic proteins to new sites of CME. And these are largely adapter proteins that bind to cargo at the plasma membrane, as well as lipids, and recruit later arriving factors in the endocytic machinery. These are followed by actin modifiers and regulators that help to generate force that pushes away from the plasma membrane, helping to form these endocytic invaginations in the plasma membrane, which are eventually released into the cytoplasm as clathrin-coated vesicles with the help of these scission factors. And those coated vesicles are eventually uncoated so they can be delivered to and fuse with early endosomes, shown here. Now, my project focuses largely on these middle coat proteins shown here and the adapters in this group of proteins. There are a number of different classes of endocytic adapter proteins shown in different colors here, and it's important for the cell to have a large variety of adapters so that it can respond to changes in the environment. For example, perhaps the cell would want to leave a certain type of cargo protein at the plasma membrane while internalizing others in response to some change, and adapters allow for that to happen as they recognize specific sequences in the cytoplasmic regions of these cargoes. And the work I'll be telling you about today focuses on the CLASP family of adapters for clathrin-associated sorting proteins. And we're going to talk about the epsins first, which in yeast, which has two epsins, are called N1 and N2. And these are an essential gene pair, so deleting both of them from the genome makes the cells inviable. This is what we call a synthetic lethal interaction. And to just kind of walk you through some of the features of these proteins, you can see that they have a similar set and arrangement of domains and motifs. They have an N-terminal nth domain for epsin N-terminal homology that binds to the plasma membrane, ubiquitin interacting motifs that associate with cargo at the plasma membrane. MPF motifs bind to EH domains that are common in other endocytic proteins. And finally, they have these clathrin binding motifs at their C terminus. Now, I mentioned that you can't delete both of these genes in combination, but if you leave behind just a single nth domain from one of the two epsins, you get our double delete plus nth strain. And these cells are viable, and they don't really show any significant endocytic phenotypes. So what you're looking at here is the localization, the steady state localization of a cargo protein, sterol3, which here is tagged with GFP. Sterol3 is a mating pheromone receptor in yeast that when there's no mating pheromone around, after being delivered to the plasma membrane, is constitutively endocytosed and delivered to the vacuoles, which is what you're seeing here. And that's basically what we see in the double delete plus nth cells as well, most of the signal within the vacuole. Now, there are two other similar adapters in yeast. These are the AP180 homologues, YAP181 and 2. And you can see that they're similar as well to the epsins. They have an nth domain, which is not essential for viability of the cells. So you can delete both of these adapters in this double mutant context as well, creating what we call our quad delete plus nth strain. And this is where we start to see endocytic phenotypes. So unlike the double mutant, the quad delete now shows sterol3 accumulating at the plasma membrane at the surface of these cells and highlighting their outer edge. And I want to mention that reintroducing any of the four full-length adapters into this quad delete strain can rescue the endocytosis phenotypes that we observed. So here is reintroduced ENT1 or YAP181, and you can see that most of that sterol3 cargo is now localizing to the vacuole instead of concentrating at the plasma membrane of these cells. And this quad delete plus N strain provides an ideal background for trying to identify other factors involved in CME or perhaps components of parallel endocytic pathways. And so what we did in this project was utilize that quad delete plus N strain to try to identify mutations that are lethal in this background. So synthetic lethal interaction where on their own the cells would be viable, but in the context of our endocytic mutant, they make the cells inviable. And so in order to do this, we used a colony sectoring assay that takes advantage of an interesting phenotype of adenine synthesis mutants in yeast. So what we did was we took our quad delete plus nth cells and introduced a plasmid that contains the full-length N1 gene and also turns the cells red. So any of the cells that have the plasmid are red, whereas any that don't are going to be white. And we treated those cells that we grew on media to select for the plasmid with the mutagen ethylmethane sulfonate to introduce point mutations. And from there, you get two possible scenarios. Either you've introduced a synthetic lethal mutation that now means that the cells require that full-length ENT1 containing plasmid for viability and they stay red, or you've introduced either no or non-relevant mutations. The cells can lose the plasmid and they turn white. And these two different scenarios look different when you grow these cells on a plate. So whereas these non-relevant mutants, they produce these sectoring colonies where as cells divide, they can still grow after losing the plasmid, and that cell and all of its progeny will turn white, creating these white sectors. Whereas any synthetic lethal mutants that lose that plasmid will not be able to grow on the plate. So the colonies that form are composed entirely of cells that have retained that plasmid, and they're completely red, non-sectoring. So we isolated a large number of these non-sectoring mutants, and with the help of a number of other people, conducted several tests to confirm that we were working with the types of mutants that we wanted to be working with. For example, we confirmed that the non-sectoring phenotype was not due to integration of the plasmid into the yeast genome, that these were caused by single mutations. We identified the recessive mutants and grouped them into different complementation groups. And from that, we were able to identify 8 multi-member groups of mutants that in theory would have mutations in the same gene. And I'm going to be telling you about one of those groups today, in which we were able to rescue the cells with 2 overlapping intervals of the yeast genome, which both contained 3 full-length genes. We kind of homed in on VPS4 because it's involved in that endocytic pathway that I showed earlier, specifically in forming these multivesicular bodies or late endosomes. And it does this by disassembling what are called escort complexes, these groups of proteins that act at the surface of endosomes to move cargo and endosomal membrane into the lumen of those organelles. And VPS4 and its complex of proteins specifically disassemble the escort-3 complex. So there's an escort 0, 1, 2, and 3. VPS4 acts on this last complex, disassembles it, releasing those proteins for further rounds of this process and allowing for the release of that vesicle into the lumen of the endosome. Now, VPS4 mutants have a really distinct cargo trafficking phenotype. So what you're looking at here is the localization of a different cargo this time, fluorescently tagged carboxypeptidase S or CPS. And you can see that in wild-type cells, CPS localizes mostly to the lumen of vacuoles. So within that organelle, this is outlined by this lipophilic dye, FM464. But in the VPS4 delete that's lacking this protein, the cargo instead is concentrating at the vacuolar limiting membrane and not making it into the vacuole itself. And also that it's accumulating in these adjacent compartments called class E compartments, because VPS4 is what's called a class E vacuolar protein sorting mutant. And so we wanted to perform an experiment to see if our mutants, our synthetic lethal mutants, had similar phenotypes in terms of cargo trafficking. And so we took representatives from all 8 of our complementation groups, not just the group that we thought might have mutants for VPS4, which is group 3 here. But indeed, we saw that this group of representative mutants, this group 3 set, did have what looked to be a class E cargo localization phenotype of GFP CPS, unlike the wild type and quad delete plus nth strain alone. And interestingly, though we're not going to focus on these, many of the other complementation groups showed abnormal cargo localization as well. Some even looked class E as well. So that's interesting and might indicate that vacuolar protein sorting mutants are commonly lethal with endocytic mutants when you combine the two. So we sequenced the gene in those two representative mutants from complementation group 3, and indeed identified mutations in the VPS4 gene sequence. One was an early frame shift mutation that led to a stop codon near the N-terminus of the protein, and another was a missense mutation at residue 283 from aspartic acid to valine. And so after getting this result, we wanted to see if we could delete the VPS4 gene from our reporter strain and get that same non-sectoring phenotype. And so that's what we did in this experiment I'm showing here. We've got a parent strain that is able to sector on this rich non-selective media, a positive control for the non-sectoring phenotype, which was a synthetic lethal mutant we identified and isolated in our screen, and then a parent strain in which we deleted the VPS4 gene. And you can see that removing the gene from those cells makes the colonies that they form completely red and non-sectoring. So it does look like VPS4 becomes essential in our quad delete plus nth context and makes them require this full-length N1 for viability. And we also grew these cells on this counter-selective medium that contains 5-FOA. And this selects against this plasmid here because it contains the UR3 gene. And so these cells, like the synthetic lethal mutant, don't grow on this counter-selective media, just indicating in a different way that VPS4 is indeed synthetic lethal with this background. And next, we wanted to see if it was the activity of the large ATPase domain of VPS4 specifically that's necessary for the viability of our endocytosis mutants. And so we utilized this VPS4 E233Q mutant, and that point mutation inactivates the large ATPase domain of this enzyme. And there are a lot of controls here that I'm not really going to get into just for the sake of time, but they looked correct to us. And the most important one for our purposes is this one on the far right, in which we introduced that VPS4 mutant into our quad delete plus nth cells that also lacked their original VPS4 gene. And it looks like on another type of counter-selective media, these cells are unable to grow. So even with the protein there, the inactive form is not sufficient for viability of these cells. And I want to mention that this experiment was performed by one of my undergraduate research assistants, Sanjana Pasari. And so having identified that VPS4 is essential in our quad delete plus nth cells, we next wanted to determine if maybe other members of the VPS4 complex or perhaps certain escorts are also essential in this context. And so similar to the VPS4 mutant that I made in our reporter strain and that I showed before, I also made mutants for over 20 of these escorts and VPS4 complex components, including the 3 that I'm showing here. SNF8 and VPS36 are escort 2 factors, and SNF7 is part of the escort 3 complex. And I'm showing these 3 because these were the ones that we identified as also being essential in our quad delete plus nth strain. And so you can see that the colonies that they form are non-sectoring, they're completely red, and these cells do not grow on our counter-selective 5-FOA media. So this indicates that it's not just VPS4, it's other escorts, and in fact, the MVB pathway that all of these proteins help to mediate that are important for the viability of our endocytosis mutants. And we also wanted to figure out if it's an interaction between VPS4 and our quad delete strain specifically that we're observing, or if perhaps there are more interactions between a VPS4 delete and other mutations that disrupt clathrin-mediated endocytosis. And so I created a double mutant that lacks both the VPS4 gene as well as END3. END3 encodes another CME protein. It's not an adapter, but it's also really important for this process. You can see that in these END3 delete cells, sterol 3, that cargo we talked about before, concentrates at the plasma membrane. So we see similar endocytic defects. And in all of these strains, the single mutants and the double mutant, I transformed them with a full-length End3 cover plasmid, similar to like what we did for the quad delete with the full-length End1. And these cells were transformed successfully. They're growing on selective media that lacks uracil. And the single mutants grow just fine on our counter-selective media, so they're able to lose this N3 plasmid, but the double mutants don't grow on media containing 5-FOA. And so it looks like this combination of mutations is lethal as well, suggesting that the loss of VPS4 has an interaction with the disruption of CME in general and isn't specific to our quad delete plus nth background. And so that brings us to the end of the first story that I wanted to tell. The next one has to do with why this interaction exists in the first place. Why is MVB formation seemingly necessary for the viability of our CME mutants? And we tried a number of different things. For example, we tested the ability of ubiquitin overexpression to rescue these cells. We thought that maybe ubiquitin levels were depleted in this context below a critical level. But we kept coming back to this idea that maybe it has something to do with the phenotypes that we observe in our quad delete strain with this increase in the amount of cargo proteins at the plasma membrane. And maybe there's something similar going on in our VPS4 delete cells alone. And so in order to test this, I made strains that express sterol3 GFP, that same cargo, in both wild type background and in VPS4 delete cells. And we noticed that similar to our endocytosis mutants, there seemed to be an increase in the amount of sterol3 GFP at the plasma membrane of these cells compared to wild type. And when we quantified the amount of sterol3 at the surface of these cells, we found that indeed there was a statistically significant increase in the amount of the cargo at the surface of these cells for some reason. So we tested some of the non-lethal escorts that I had made, those mutants, as well as some of the mutations that were synthetically sick but not totally lethal. The non-lethal group, by eye and after quantification, showed no real difference compared to the wild type, whereas the synthetically sick cells may have showed some increase in the amount of sterol3 at their surface, particularly VPS36. So it's possible that there is a continuum from wild type to VPS4 in terms of the amount of cargo that ends at the plasma membrane and how sick the cells are when you delete these genes from an endocytosis mutant. And we noticed that this was not specific to sterol3. We tested two other cargos, MEP3, which is an ammonium permeasing yeast, as well as PTR2, a di and tripeptide transporter. And like sterol3, the cargos here that were fluorescently tagged seem to accumulate at the surface of these cells more so in the VPS4 delete, and upon quantification showed that this difference was statistically significant. So it's not something that's specific to a particular cargo, but maybe something more general to protein trafficking in these cells. And so next, we wanted to try to figure out why we were seeing this increase in protein cargos at the surface of VPS4 delete cells in particular. And we thought that one reason might be that perhaps cargos that aren't making their way into the lumen of multivesicular bodies and late endosomes may instead be getting redirected into recycling pathways, as they accumulate on the surface of endosomes. And so in order to test this, we took advantage of a component of the endocytic recycling pathway in yeast, RCY1, and I generated strains that were double mutants lacking both VPS4 and RCY1. Now, the RCY1 delete strain alone appeared similar to wild type in terms of sterile 3 GFP localization. But the double mutant seemed to block that accumulation of sterile 3 at the plasma membrane that we see in the VPS4 delete alone, and instead accumulates in these amorphous compartments within the cell. Not much is at the surface of this double mutant strain. And we're not exactly sure what these compartments are. Another of my undergraduate research assistants, Tony Yao, has been working to try to figure out what these are. And in one experiment he did, he stained these double mutant cells with vacuolar dye CMAC and looked at the localization phenotype compared to the sterile 3 GFP cargo. And he noticed that these cells do indeed have vacuoles. They may be more fragmented, but they do not co-localize. That stain does not co-localize with our sterile 3 GFP. And so we're still trying to figure out where the cargo is ending up in these cells. It's possible that these are exaggerated class E compartments, or perhaps enlarged early or cycling endosomes, although it's possible that they may even be enlarged trans-Golgi networks, since there's some recent evidence that yeast may have a more simple endomembrane system where the trans-Golgi network takes on the responsibility of the early and recycling endosomes and doesn't have those organelles at all. I made strains with other fluorescent cargoes, again, MEP3 and PTR2, just to see if we observed similar phenotypes, and indeed, these cargoes don't appear to accumulate as much at the plasma membrane of these cells as in the VPS4 mutants. So again, this seems to be a more generalizable phenotype of cargo trafficking in these double mutants, not something that's specific to one cargo. And we quantified the amount of these cargoes at the plasma membrane. I'll just quickly show this here and mention that for all of the double mutants in white for each of these 3 different cargoes, we saw that there was a significant decrease in the amount of the cargo at the plasma membrane of the double mutants compared to the VPS4 delete strain for each of the 3 cargoes. So it does look like it's endocytic recycling of internalized protein back to the plasma membrane that's causing this increased cargo concentration at the cell surface that we observe. And it looks like these VPS4 delete cells really do rely on the endocytic recycling of protein in order to sustain some of their normal phenotypes, because if you delete RCY1 from a VPS4 delete strain, those cells, those double mutants, then become temperature sensitive. Yeast like to grow, they grow optimally at 30 degrees Celsius, and the double mutant strain is growing fine here on this rich media. But at 37, they're no longer able to grow like the single mutants. And a final undergraduate research assistant, Pedro Castanera, performed or generated growth curves for each of these 4 different strains and found that there is a significant lag in the growth curve for the double mutant compared to any of the other 3. And so these cells really do have some significant growth phenotypes. And so this brings me to our model as to what we think might be going on here. So in an endocytosis mutant, you've got protein accumulating at the plasma membrane. And if you add on a VPS4 mutation and disrupt that MBB pathway, it's possible that it's this combination of decreased endocytosis and more cargo being misdirected into endocytic recycling pathways back to the plasma membrane that overloads the cell surface with protein. And perhaps it's specific proteins like specific amino acid transporters. Perhaps it's an overabundance of misfolded proteins that disrupt the integrity of the cell surface. Or maybe it's even just kind of a general overloading of protein at the cell surface. And so we're still trying to figure out what's going on there, but I hope some of the ideas you take away from this talk are that the MBB pathway that includes VPS4 and escorts is essential in endocytosis mutants, that disruptions in one membrane trafficking mechanism can have consequences for adjacent pathways, and the disruption of the MBB pathway, such as with VPS4 mutants, can have effects on the composition of the plasma membrane. And so, what are some of the implications of this for SARS coronavirus 2 and the difference in severity of COVID-19 cases, particularly the elderly, those with pre-existing conditions, and men to some extent being more susceptible to dying from this disease? For one, the levels of ACE2 may vary in these different populations. For SARS coronavirus 2 to enter a cell, it first has to attach to it through the binding of its spike proteins to ACE2. And a difference in ACE2 concentration at the plasma membrane could determine a person's susceptibility to infection or their risk of having a severe form of the disease. And so an appealing hypothesis would be that maybe ACE2 levels are higher in these groups, and that the increased presence of ACE2 would make them more susceptible to this novel coronavirus. However, it's been shown that ACE2 levels actually decrease with age. So perhaps the real effect here is that there's a limited amount of ACE2 on the cells of older adults, and this is further diminished by infection with the virus, which induces the uptake of ACE2 by endocytosis after its attachment. And this may decrease the level of ACE2 at the plasma membrane to critical levels. And ACE2's normal function is to convert angiotensin 2 to angiotensin 1-7. Angiotensin 2 is associated with vasoconstriction and an increase in blood pressure, inflammation, things like that, whereas angiotensin 1-7 is associated with vasodilation and a decrease in blood pressure. So it's possible that the increase in the concentration of angiotensin 2 as a result of this lack of sufficient ACE2 might be leading to vasoconstriction, hypertension, and the inflammation that's seen in these more severe cases, and potentially why people with cardiovascular disease and hypertension also have higher fatality risks. And additionally, you've probably also been reading about what are sometimes called cytokine storms in patients infected with SARS coronavirus 2. Cytokines are any number of small proteins secreted by specific immune cells, and these are recognized by receptors on target cells throughout the body. And so normally this is a good thing and helps the body to recover, but during these cytokine storms, also known as cytokine release syndrome, a large number of leukocytes are activated to release these cytokines, which in turn activates other leukocytes to do the same. And this process is accompanied by inflammation. And so instead of helping with the recovery, it actually leads to a deterioration in the patient's condition. And so perhaps this response is not only due to the increase in concentration of cytokines themselves, but maybe also an increase in the concentration of the receptors for those cytokines on the plasma membrane of target cells. And another point I want to make before moving on is that a recent study showed that the actual entry event where SARS-CoV-2 fuses its viral membrane with the endosomal membrane to allow for the release of its genome into the cytoplasm, that this event occurs within late endosomes specifically, not early endosomes. And you'll remember that the formation of late endosomes depends on the MBB pathway, on the activity of escorts and VPS4 to form those intraluminal vesicles that are hallmarks of this organelle. And so it's possible that disruption of the MBB pathway may make it more difficult for the virus to infect cells. But it's not really a trade-off that I would want to make, since the normal functioning of this pathway is critical for the health of cells in an organism. But it's possible that disrupting this transition to late endosome could impede viral infection. And our findings also have potential implications for uncontrolled cell growth. Cells can divide in response to growth factors that bind to receptors on their surface and signal for the cell to divide. And interestingly, one of the escorts in mammalian cells is called TSG101, short for tumor susceptibility gene 101. And researchers found that mutations in this gene can predispose cells to becoming tumorigenic. And this is likely due to another phenotype that they observed, that there's an increase in recycling of epidermal growth factor receptor in this context. And so it's possible that mutation in other components of the MVV pathway, or maybe even most components, would have a similarly detrimental effect and possibly predispose cells to becoming cancerous. And so again, hopefully you'll come away from this webinar with the idea that disruptions in one membrane trafficking pathway can have effects on adjacent mechanisms and thereby lead to severe consequences, not only for cells, but the organisms that they're in. And with that, I'd just like to quickly thank all of the current and former members of the Wendland Lab who helped with this project, particularly my advisor, Dr. Beverly Wendland. I'd also like to thank our collaborators, Dr. Carolyn Norris, and the students in her genetics lab course who helped with the initial screening for synthetic lethal mutants, Dr. Derek Prosser at Virginia Commonwealth University. And again, I want to thank the organizers for giving me this opportunity to share with you today. And quickly before you go, I'd just like to mention that Dr. Wendland will unfortunately be closing her lab at the end of the spring. So I'm currently on the job market. If you enjoyed this webinar, please feel free to reach out to me on LinkedIn to connect and or discuss potential opportunities. So thank you all for watching and stay healthy.